Good afternoon, everyone. I am Jenita Thinakaran. I presently work at USDA-ARS with Drs. Muninesa, Dr. David Horton, and Dr. Cooper. Today's presentation is from my PhD dissertation research that I worked in Dr. Henne's lab. I miss Dr. Henne here, so I'm presenting today about the assessing preference of the potato psyllids, how they prefer different solanaceous among the different solanaceous hosts, and also between infected and the free yellow-free host plants. Broad outline of my talk will be a brief introduction, and I will move on to my research objectives, the experiments I conducted, conclusions based on my results, and scope for future research. In this disease triad, there is constant interaction between the vector, how the vector prefers the host plants, and the effect on the vector by carrying the pathogen, and also interaction between how the presence of or absence of the pathogen affects the host plants. My research objectives as part of this experiment were to evaluate preference among the different solanaceous hosts, and also to test preference between the LSO infected and LSO free hosts. The solanaceous hosts I tested were used in my experiments were potato, tomato, pepper, eggplant, and a wild solanaceous host, the silverleaf nightshade. Silverleaf nightshade is widely prevalent in Texas, especially around potato growing fields. We have observed this. A psyllid monitoring program, we had sticky traps in a silverleaf nightshade patch around potato field, and we collected adults. We sampled the leaves, and we found nymphs, eggs, and all instars of nymphs, so which indicates a breeding activity going on in the silverleaf nightshade patch. Also, I did some biology and life history studies of the potato psyllid on silverleaf nightshade in comparison with the potato as a main host. Based on the life table parameters, I estimated the life tables, and what we found was potatoes to be a superior host in providing in the nutrition provided by silverleaf nightshade was inferior. However, the effects of LSO did not make any difference on the growth and development of the potato psyllid. Coming to the first aspect of my talk, selection of the preference of potato psyllid for different host plants. This is the interaction between the vector, how it prefers the different host plants. These 5 host plants were used in pairs, were planted in pairs, making 15 possible comparisons. The trial was replicated 4 times, and 2 field trials were conducted. The field population psyllids were assessed at weekly intervals throughout the growth stages of potato. The results from these trials, the first field trial, what we found was potatoes were highly preferred starting from the growing season, and followed by tomato, and then pepper, eggplant, and silverleaf nightshade. Similar results were also obtained from a second trial, field trial. Potatoes were highly preferred, equal to tomato, followed by pepper, eggplant, and silverleaf nightshade. I also assessed how this tried to correlate how this different growth patterns correlate well with psyllid abundance. At the time of planting, when the potatoes were still sprouting, tomatoes were already planted. We found this initial stages, tomato had more psyllids, and as the potatoes started establishing in the field, psyllid abundance was on potato plants. Finally, when potatoes gave way and started senescing, psyllids started moving to other host plants in the absence of its favored host, started moving to other host plants, including silverleaf nightshade. So I did some laboratory experiments to test the effect of the plant size on the psyllid preference. I used a large potato plant and a small potato plant. Psyllids continuously preferred the large potato plant. This I used as a control, 2 potato plants of the similar size, and there was no significant difference throughout the study. I went on to use potato and tomato plants. I used a large potato plant and a small tomato plant, and a large tomato plant and a small potato plant. Irrespective of the host species, the psyllids preferred to settle on the larger of the 2 hosts. When I used the same size tomato and a potato plant, there was not any difference. This is comparing among the tomato plants, a tomato large and a tomato small, a potato large and a potato small. Coming to the next aspect of my talk in assessing preference for LSO-infected and LSO-free hosts. This involves the interaction between the infection of plants with this pathogen and how the psyllid prefers the infected or the uninfected plant. Plant 3 to 4 week old plants were infected with this LSO using about 7 psyllids per pouch and leaving them for a week. Pouches were removed along with the leaves, and the plants were allowed to disease progression for 3 weeks, 6 weeks. Later, they were tested, they were evaluated for preference. This is the status of the infected plants. Of course, Ven showed some pictures. Here the LSO type is B, and the psyllids are central psyllids. Potatoes did not stay longer. 3 to 4 weeks later, potatoes started wilting, and they started dying. They didn't stay longer. Tomatoes were pretty robust. 3 weeks later, they didn't show any, they were asymptomatic. 6 weeks later, some kind of curling, and 8 to 9 weeks later, they started wilting. Eggplants and peppers were very robust. There was no visible symptom at least 7, 8 to 9 weeks later. Silverleaf nightshade did not show any visible symptoms, but 4 weeks later, possibly due to pot-bound conditions, they started dropping their leaves, and this happened also in the control plants. Experiments were set up in the laboratory. The treatments I used, the main treatment was comparing LSO-free with LSO-infected plant. That was a treatment. And the controls I used in the experiment was comparing between 2 LSO-free plants and using 2 LSO-infected plants. I used both LSO-infected psyllids as well as LSO-free psyllids. I released in separate experiments. I really used both these psyllids. And host plants I tested: potato 1, 2, and 3 weeks after infection, tomato, eggplant, and peppers 3 and 6 weeks after infection, and silverleaf nightshade 4 weeks after infection. So that's in total, there were 20 experiments in all, and I have summarized the results from the 20 experiments, bringing in only information from the uninfected and the infected host pair. The other control treatments I eliminated. There was no significant difference. And among these, in all these experiments, I didn't find a lot of significant difference between the LSO infected. The psyllids settled uniformly on the LSO infected as well as the clean plants. Only in a few of these experiments did I find significant differences. And that too, more of the psyllids prefer to settle on the infected plants. The difference column indicates the difference in the number of psyllids that settled on the uninfected versus the uninfected. This is the first versus the second plant. A positive difference indicates that the more psyllids settle on the first plant, the clean plant, and a negative difference indicates that more psyllids settle on the infected plant. In order to graphically represent, I also represent this graphically in potato. In 1, 2, and 3 weeks, this is the The y-axis shows the number of psyllids, and the x-axis shows the number of in hours. Up to 70, I recorded this observation up to 72 hours after release. And in all this, I have found continuously more psyllids prefer to settle on the clean plants, the uninfected or the LSO-free plants. In these cases, no significant differences were found. Okay, here, the error bars are based on individual settling point. However, the significance is based on repeated measures ANOVA over the entire period of time. A similar trend was also observed on tomato plants. In most cases, there was no significant difference. However, in this situation, there was psyllids settling more on the uninfected clean plants. There is one trend here which was very close to significance, where the LSO-free psyllids tend to prefer the LSO-infected plants. This was the only situation where psyllids settle more on the LSO-infected plants. So that was the only thing, but however, since it was not statistically significant, and in most cases we observed that the psyllids preferred equally to settle on both the infected and the free plants. Silverleaf nightshade, on the other hand, did not exhibit any visual symptoms, and they were totally asymptomatic. 4 weeks after infection, psyllids settle uniformly on both the infected and the uninfected plants, both in case of LSO-free infected psyllids and LSO-free psyllids. We didn't find much difference on using silverleaf nightshade either. The infected silverleaf nightshade plants were transferred to the field and were retained for about a year, and we tested samples of roots and shoots, and they were found to retain LSO throughout the summer. We also conducted psyllid acquisition studies. We allowed clean psyllids to feed on these plants and removed them, and the psyllids were tested and found to be LSO positive. And we confirmed that silverleaf nightshade is a possible pathogen reservoir host in the lower Rio Grande Valley. To summarize my talk, the psyllids moved to silverleaf nightshade in the absence of its favored host. Plant size could be a confounding factor in experiments with preference. Symptom expression with LSO varied from plant to plant and also from the host species. Potatoes were highly sensitive, they were susceptible. Tomatoes were fairly robust. Eggplants and tomato were highly robust. They exhibited symptoms very later. They stood the test of time. Psyllids, in most situations, they prefer to settle on both LSO-infected and LSO-free plants. Most of them were, the plants were asymptomatic. In some cases, they preferred LSO-free plants. So based on these studies, I could say psyllid settling has a specific preference. They did not settle on any plant just by chance. There is some mechanism they exhibit in order to locate their host plants. So further investigation into mechanisms of host finding needs to be done, whether it's visual or olfaction needs further investigation. Olfactometer studies and studies under dark conditions could reveal differences, could tease out the differences between these olfaction and the visual stimuli. With this, I'd like to close my talk, and I thank the members of the IPM lab for helping me with the conduct of the experiments, and I thank you all.